So thank you everyone for joining the PFAS Community of Practice. Today we are going to hear from Andreas Cardenas and Allison Ehrlich from Stanford and UC Davis respectively. And without further ado, we will let Andreas kick off. Thanks, Andreas.
Thank you, Fiona. So yeah, welcome everyone. So today we're going to be doing brief talks on the immunological effects of PFASs. And particularly, I'll be going over some of the Superfund project that I'm leading. So I'll briefly introduce myself. I'm Andres Cardenas. I'm an assistant professor of epidemiology and population health at Stanford, the medical school. However, I've been involved with the Superfund program at UC Berkeley now for a number of years. For this cycle, I lead one of the projects and also help with the data management core as well. So I wanted to kind of briefly go over some of the proposed mechanisms. This is definitely out of my area of expertise, but hopefully will kind of elicit some conversation among us. This is a literature review that was performed in 2023, looking at different mechanisms of immunotoxicity for PFAS. For a long time, some of the toxicological and epidemiological studies have shown that PFAS are immunotoxicants. And this literature review performed, about a year ago, showed that there's multiple pathways. Some of them are inducing immunotoxicity. There's inhibition of different immunological pathways, and there's also modulation. But you see a lot of gray here, which is unknown potentially. So it's a potential pathway with the dashed line. So at least the conclusion for this is that PFASs, and broadly speaking, usually we're talking about PFOA and PFOS that have been historically studied, induce inflammatory processes. They also alter cytokine production, and that's one of the things that we have looked at in our project. They also interfere with the processing and presentation of antigens for cells, and then they also modulate cell population. And interestingly for us, the antibody production as well following vaccination. So that's something that we're looking at and very interested in. But the picture, at least for me as an epidemiologist here, looks fairly complex. So I'd love to have more conversation with the toxicologists as to whether we're arriving at some common mechanisms potentially across the vast sea of PFASs. So this has been over a decade now. This is a seminal paper in which Dr. Philippe Grandjean showed in the Faroe Islands that both PFOS and PFOA are associated with decreased vaccine response following vaccination. Particularly, he was able to show that pregnancy or prenatal PFOS decreased diphtheria antibodies, about 39% decrease in the concentration of antibodies following vaccination, with increase in exposure. So this is not trivial, but clinically important. Cross-sectionally or more, towards childhood at age 5, he was able to observe that both PFOS and PFOA exposure decrease antibody production toward tetanus and diphtheria. And then at age 5, exposure for PFOS and PFOA, he observed greater odds of falling below the clinically protected levels for both tetanus and diphtheria as well. So this was the basis of a lot of the risk assessment that was performed early on as a sensitive population, both children and pregnant women early on. So that was based, a lot of the risk assessment that was performed, quantification was based on this study from humans. Since then, there have been multiple studies. People have been interested in different vaccines with different different antibodies with different study designs as well. So this is a meta-analysis of 14 studies, 13 of which were unique. And you can see here that the most widely studied PFAS as well, again, PFOA and PFOS, showing that PFOA decreased antibody types, and this is across all antibody types. So you can see here color-coded on the right, like hepatitis, HPV, as well as measles, mumps, and rubella and tetanus have been historically studied, 5% decrease, and this is in the absolute quantification of titers. The most consistent findings, again, come from diphtheria, rubella, and tetanus that have been most supported, at least for PFOA. The associations with PFOS were a little bit more imprecise, also going on the negative direction for the meta-analysis. One current limitation is that, again, we have only been limited to looking at 5 PFASs, the traditional 5 or 6 that we study, that we can measure in plasma. And we have not looked at mixtures, so that's one gap that we're looking at right now. So for project 2 of our Superfund program, we're looking at early life exposures, both to PFASs, but also metals, which are very common contaminants. We're looking at cytokine production as a biomarker of immune development and maintenance. And we're also looking at vaccine titers, Tdap and MMR at age 7. We also hypothesize that epigenetic programming that happens very early during fetal development might mediate this association. So that's one piece of the puzzle that we would like to try to explain how this happens. How does an exposure early in life, like prenatal, might predispose you to have lower vaccine response later in life. So it has to be some biological explanation as to why this might happen, and epigenetics might provide a good theoretical model to study this or foundation. So this is results from our first kind of aim to look at cytokine production in cord blood. So this is following the measure of prenatal PFASs. We observed that mucosa specifically was associated with multiple immune response biomarkers, so graded an increase in lymphocyte proliferation response after stimulation of the cord blood with egg antigen. We also observed lower odds of having INF alpha, INF gamma detected in response to dust mite antigen presentation in the cord blood again. So these were experiments to measure the response to these antigens. And then we also observed increase in lymphocyte proliferation after cockroach antigen stimulation, and that was done in the mixtures model. So we have some evidence that they act together as well. And this is, a prenatal exposure influencing these immune biomarkers in the cord blood at delivery in the infants. So we have moved on to look at antibody response titers. Consistent with what others have found, we also found that different types of PFAS, not exactly PFOA like was reported in the Faroe Islands, but ethylated fossa and MIFOSA were both associated with lower pertussis titers. And both MIFOSA, PFNA, PFOA, and the PFAS mixtures, all of them were associated with higher risk of not having a detectable or borderline MUMS titer at age 7. So this is important, as we know infectious diseases are kind of making a comeback. So trying to understand environmental contaminants that might influence this association is pretty important. And we also have some evidence that at least some of the metals, they were not co-interacting, they were not jointly interacting with each other, but some of the metals might increase the risk of having non-detectable measles titers. But we were able to account for that in our analysis. So with that, I think I'm at my 7 minutes. I wanted to briefly show our results and elicit some discussion as well. So with that, I think I'll pass it on to Allison, and I think we have questions towards the end, correct, Leona?
Yeah, that can work.
Okay, thanks.
Okay, great. That worked really well because I was able to delete a couple of my slides. Let me just share my screen.
Looks good.
That looking okay?
Yeah.
Okay, great. Okay, so I am Allison Ehrlich. I am an assistant professor at UC Davis in the environmental toxicology department, and I'm going to talk about some of the work that we've been doing in vivo and vitro to assess the immunological effects of PFAS, particularly the non-legacy PFAS. So as Andres already nicely went over, and I'm not going to go over in too much detail, the legacy PFAS, particularly PFOA and PFAS, there's a lot of data showing that these are presumed to be immunotoxic, and this comes from the MTP monograph. This is from 2016, so there's more studies since then. Andres nicely summarized, but mostly showing that PFOA and PFAS are immunosuppressive, particularly to the antibody response, and that comes from both animal and human studies showing reduced vaccine antibody titers in individuals exposed to higher levels of PFAS and PFOA. So what we were interested in is what about all the other thousands of PFAS chemicals? And we started with this read-across approach. This was done, this was led by a collaboration and a grant that where Michelle Amaro is the PI, along with Martin Smith and collaborators at UC Berkeley, where we started with this list of 160 PFAS chemicals, and this list was compiled from chemicals that were of interest to OWIHA, the Water Board, and also a manuscript by Hook et al. And we did cluster, I didn't do this, this was led by the Berkeley group, but cluster analysis to try to find common structures that were to try to identify commonalities between these structurally diverse PFAS. And we ended up with about 25 clusters, and we were specifically looking to do this read-across approach in clusters that had at least one member that there was some evidence of immunotoxicity, one member that had a shorter chain chemical, and one member that was under characterized, so that there wasn't really any published data in PubMed or other resources about their immunotoxicity. And we took two approaches to assess immunotoxicity. The first was an in vivo approach. And we did this really classic immunoassay called the T-cell dependent antibody response assay, where you pre-expose, in this case, mouse, to the xenobiotics, so the PFAS that we were interested in, injected them with sheep red blood cells, which are T-cell dependent antigen. The antigen presenting cells, the APCs, so these are our macrophages and dendritic cells, take up the sheep red blood cells, present them to T cells, and because it's a T cell dependent antigen, B cells only produce antibodies against the sheep red blood cells when there are functional T cells, which require antigen presentation by APCs. So it ends up being a nice assay to look at both the innate immune response, T cell responses, and B cell production of antibodies. The first cluster that we looked at was composed of Gen X, which had previously been shown to have some immunotoxic potential, along with this chemical, which I'm just going to abbreviate as HFPO-TA and HFPO-TEA. And during this first cohort, we wanted, because these haven't really been studied, we wanted to assess tolerability and also look at the immunosuppressive potential. Unfortunately, what we found was that HFPO-TA and HFPO-TEA were incredibly toxic. So there was just acute toxicity associated with weight loss that almost reached our ICOOK endpoint. So we had to reduce the dose to 1 to 10 to keep the study going. But unfortunately, it still wasn't enough, and mice continued to lose weight. Our main toxicity outcome that we saw in these mice were actually not immunosuppression, but major hepatotoxicity. So there was some really interesting growth pathology that Michelle Amaro's lab is going to be looking into at more detail. But we saw hepatomegaly and kind of this lipid accumulation phenotype that looked pretty interesting that we are going to do more studies on. But in terms of a reduction of sheep red blood cell antibody responses, we didn't really see any pronounced outcome of immunosuppression. Because of the acute toxicity that we saw in the first study, we had a second cohort of mice where we repeated our studies, but with 3 doses for HFPO-TA and TEA. So 10-fold and 100-fold lower dose, and we included dioxin as a immunosuppressive control. And again, what we saw is this dose-dependent increase in hepatomegaly, but we didn't end up seeing any changes, any significant changes to the antibody response. I'm just going to fast forward a little bit, and we ended up kind of switching gears to see if we could do a larger screen of immunotoxicity of these PFAS chemicals. So we shifted to this human macrophage assay where we differentiated monocytes into macrophages and stimulated them with a TL4 agonist and looked at pro-inflammatory cytokines and looked at the same longer chain PFAS compounds that we used in our in vivo study, but also added a variety of short chain PFAS compounds as well. And really what we found from these in vitro assays that we're still actually are ongoing, but this is some preliminary data, is that while we saw toxicity with the longer chains, so our HFPOTA and PFOA and PFAS, the short chains didn't really induce any gross cytotoxicity. And similarly, we found the expected reduction in TNF-alpha with PFOA and PFAS, but not the shorter chain chemicals. And same thing with IL-1 beta. So maybe was not overly surprising, but that the longer chain PFAS chemicals that we looked at showed more evidence of immune suppression than the shorter chain PFAS. And we mostly were able to note this in our in vitro macrophage assays rather than our in vivo studies. And so we might be missing, our TDAR assay might be missing some of the complexities of suppressing the antibody response, since we weren't able to recapitulate that with PFAS and PFOA, as opposed to TCDD, which is classic immunosuppressant. And so that's where we're at right now. Studies are still ongoing, but just wanted to point out in our lab, Michael Goodson, a project scientist, did most of these studies with the help from a really talented undergrad, Haroon, as well as help from a PhD student in Michelle Lamerle's lab. And so I will stop there.
Thank you, Allison. We can open up to questions if anyone has any questions. Not too many. Allison, can I ask you if those two PFAS have been studied very much?
The TA and TEA.
TEA, yeah.
Yeah, so they've been really understudied. There's not much data on either of them, especially in terms of like the immunotoxicity. There are some small studies. I'm just trying to think of what has already been published about them, but we were interested in this cluster particularly because there wasn't a lot of data, and they were similar to Gen X, which does have much more characterization studies. But these seem to be much more toxic than, or acutely toxic than, than PFAS.
Okay. We have a couple of questions from Junsu.
Yeah. Oops. Okay.
If you're talking, we cannot hear.
Yeah, Junsu, you're muted.
Oh, sorry. Yeah, it was great talk. Yeah, Allison, you mentioned one short chain, but I missed the what one short chain you were talking about.
So for our in vitro assays, we were looking at a variety of short chains. I can share my screen again.
Oh, it was not one, multiple short chain you're talking about?
It was, let's see if, so the short chains that we were looking at are here.
I see. Yeah. Okay, you have a multiple short chain there. Thank you for the.
Yeah, and so far, I mean, just in the preliminary studies,
Mhm.
we're not seeing major signs of immune suppression in macrophages, but these have just are still ongoing, so I don't want to say anything definitively.
No worry. I just curious. Hey, Andreas, thank you very much again. You mentioned that, you tested the 5 PFAS, but I saw your methyl fossa. Did you have any reason you look into those two? In addition to the NONA, the NA and the P-fossa and P4? Because I'm not quite familiar with the toxicology, so not sure if you particularly choose those metabolites.
Yeah, no, thank you for the question. And I think it simply has to do with the availability of methods. So Antonia Calafat at CDC, as you know, performed the measurements. And I didn't have the chance to mention, but this is an older cohort. So the exposures are coming from 2000, 2001 maybe. So the levels of those PFAS at that point in time, they were a little higher. So that's why we measure those PFAS specifically. And of course, now there's the emerging ones that we did not cover, but if we were to go back to the blood samples, given that these children were followed prospectively, they might not be detectable. And I think you probably better than me know that it's true for some of the samples when you go back in time, some of these replacement compounds are not found in the plasma. So that's definitely a limitation, which looking at the emerging one.
No, I don't. I initially thought you choose those two compounds because they are more sensitive to your the immune toxins test. So just curious. Thank you very much, Andreas. And Allison, thank you. It was very informative.
Thank you. Alea, we have a question from Alea.
Thank you both for your presentation. This might actually be a question for both of you, but did either of y'all happen to look at associations with glyc-A or other metabolomic biomarkers for inflammation?
I haven't. Andres, have you?
We have not. I have seen a few papers looking at PFAS and metabolomics in human cohorts, but we have not specifically looked at this. But that's a great point. Given the liver disruption for Allison, I think that's probably pretty important, right?
Yeah.
Metabolomic signatures.
We've been working on a study where we're looking at metabolomics and PFAS in plasma samples and historical samples, and we were seeing glyc-A going down, whereas the rest of, which we were wondering if that had to do with just a lack of an immune response. But it's interesting to see a decrease in an inflammation biomarker.
And Alea, is this cross-sectionally, so you measured exposure and the metabolites at the same point in time?
These are historical samples that were the lipids were done first, but then they've been frozen for a while.
Mm-hmm.
Thanks, Alea. We have a question in the chat from Martha. Did you say that in your in vivo studies, the positive control dioxin was immunotoxic?
Oh yeah, sorry if I glanced over that briefly, but we were able to show that dioxin suppressed the TDAR response. So I don't even know if I showed specifically the data for it, but suppressed the IgM and IgG response. So wanted to make sure our assay was actually working properly.
And a question from Martin for both of you. What do you think the mechanism is for immunosuppressive effect? Is it epigenetic?
Oh, that's a good question. Andres, do you want to take?
No,
I
I think this is your expertise, Allison.
Yeah, my
Love to
instinct
hear from
is,
you.
I think like the slide that you posted, I think it was like your second slide, I think did a nice job of showing potential mechanisms. And my instinct is it probably isn't converging just on one mechanism, but rather a combination depending on the timing and state, and if it's developmental exposure versus mature exposure, and at what point in the immune response the PFAS are impacting immune development response or maturation. So I don't know. I'm not really convinced that it's one mechanism. I think if it was, you probably would have already figured that out, and it's probably different for the different chemicals.
Yeah, and I think, of course, I'm biased that because I do epigenetics, but I also don't think we have the right tissue. I mean, we have the leukocytes, but, getting at maybe some of the stem cells of the bone marrow that are producing the leukocytes might be a better way. But of course, in epi studies, we cannot do that. So the other thing that, in the literature is fairly heterogeneous, even with the vaccine story, that sometimes it's not the same PFAS that is showing the decrease in immune effectiveness. So I do wonder whether it's the mixtures and, the concentration of the different PFASs and the composition of PFAS that people are exposed to. I mean, Philippe Grandjean was able to show this with PFOS and PFOA, but we and others have looked at this, and we do find somewhat of a consistent story for some of the vaccine titers, but sometimes it's not very specific to like the one, PFOS or PFOA. Might be some of the other PFASs that just happen to be highly correlated with the other PFASs. So I think we need to do a better job at characterizing the exposure of PFAS, because some of the signals don't seem to be very specific to one or two PFASs. So I'm glad, Allison, you're looking at these very specific PFAS compounds as opposed to, in epi we're very limited, but I also think the exposure profiles matter a lot.
I think you bring up a good point though, that like we're not exposed to any of these individually. And so the mixtures are probably going to be telling us a lot more informative information as well. And mixtures with PFAS and also all the other things that we're exposed to, like metals, as you mentioned.
We're at time. It doesn't look like there are any more questions. Thank you both very much for your excellent talks. Very interesting. Please join us on June 2nd, where we will hear results from state and regional well water sampling and analysis from Claire Pace, UC Berkeley, and Wendy Link from the Water Resources Control Board. So I'll send out a reminder, and we will share the slides and the recording. Thank you very much again to Allison and Andreas, and thanks to everyone for joining.
Thank you.
Thank you very much.
Take care.